Gastric secretions include acid, mucus, pepsin, and other regulatory factors. Gastric glands are tubular invaginations that are lined by a single layer of epithelial cells that extend into the gastric mucosa. They empty their products into the gastric pits and from there into the lumen of the stomach. The exocrine cells that secrete into the gastric gland include the mucus cells near the mouth of all gastric glands, which secrete a bicarbonate-rich mucus that protects the stomach from the corrosive action of acid. Parietal cells that secrete acid and intrinsic factor, which is important for absorption of vitamin B12 in the ileum, and chief cells that secrete inactive pepsinogen as well as gastric lipase. The main cells that secrete regulatory factors across their basolateral membrane in response to neural or hormonal stimuli are the gastrin-secreting G cells, the somatostatin-secreting D cells, and histamine-secreting ECL or enterochromaffin-like cells. Glands in different parts of the stomach contain different types of secretory cells. So in the body of the stomach, there are generally oxyntic glands, and the primary secretions are acid and intrinsic factor, pepsinogen, as well as histamine. While glands in the antrum are primarily pyloric glands that lack parietal cells. The parietal cell changes configuration as it goes from a resting state to an active secreting state. So in this resting or basal state, the rate of acid secretion is low. And that's because at rest, most of the hydrogen-potassium ATPases, also known as proton pumps, that are responsible for pumping acid into the lumen, are sequestered in tubulovesicles inside the cytoplasm of the cell. Upon stimulation by binding of gastrin or histamine or acetylcholine to their basolateral receptors, the tubulovesicles fuse into the canalicular membrane, resulting in this large increase in surface area of the luminal membrane, as well as insertion of the hydrogen-potassium ATPases so that acid can be secreted into the lumen at a much higher rate. Inside the cell, acid is generated through the activity of the enzyme carbonic anhydrase, which allows the combination of carbon dioxide and water to form carbonic acid, the intermediate, which dissociates to produce hydrogen ions and bicarbonate ions. These hydrogen ions are actively secreted by that luminal hydrogen-potassium ATPase, or the proton pump, in exchange for potassium. The secretion of hydrogen ions into the lumen is accompanied by the release of equivalent numbers of bicarbonate into the bloodstream by this basolateral chloride bicarbonate antiporter. This flux of bicarbonate ions into the blood creates an increase in the pH of the gastric venous blood, which is called the alkaline tide. Here we can look more closely at the complex regulation of acid secretion. So during a meal, there are hormones and paracrine factors and neurocrines that all act together to ensure adequate acid secretion. This diagram shows a basic schematic of a parietal cell that has receptors on the basolateral membrane and the proton pump on its luminal membrane. Activation of vagal parasympathetic outflow to the stomach that happens when we see or smell food and when food enters the stomach stimulates gastric acid secretion through both direct mechanisms and through indirect mechanisms. First, there's direct activation of the parietal cell via release of acetylcholine from enteric neurons, which acts on the parietal cell via muscarinic receptors. So when acetylcholine binds, it activates phospholipase C and frees diacylglycerol and IP3. It's IP3 that allows release of calcium from intracellular stores that allows hydrogen-potassium ATPases to be transported onto the luminal membrane. Atropine blocks these muscarinic receptors and prevents this direct stimulation of acid secretion. Acetylcholine also acts through indirect pathways to cause acid secretion by causing the ECL cells to release histamine. The hormone gastrin is secreted by G cells in response to the thought or smell of food, gastric distension, and the presence of nutrients in the stomach. Gastrin also acts through both direct and indirect mechanisms to stimulate acid secretion. It acts directly by binding the CCK receptors, also called gastrin receptors, which causes secretion of a small amount of hydrogen ions into the lumen through this IP3 calcium second messenger system. The intestinal hormone cholecystokinin, or CCK, can also bind this CCK receptor, but its action is much less potent than gastrin at physiologic levels. Gastrin also indirectly stimulates acid secretion by activating the CCK receptor on the ECL cells. This causes them to release histamine, and that histamine then binds to the H2 receptor on nearby parietal cells, which are coupled to adenylyl cyclase, and when adenylyl cyclase is activated, there's increased production of cyclic AMP. Cyclic AMP activates protein kinase A and causes hydrogen potassium ATPases to be transported onto the luminal membrane. Histamine receptor blockers, like cimetidine, block these H2 receptors and prevent histamine from binding. The major inhibitory control of acid secretion is decreased pH of the luminal contents. So when the gastric pH is low, there's enough acid present in the lumen, and further acid secretion can be inhibited. This is mediated by the secretion of the inhibitory factor somatostatin, which is secreted by the D cells in the gastric glands. Somatostatin directly binds to receptors on nearby parietal cells, causing inhibition of adenylyl cyclase and decreasing cyclic AMP, which ultimately decreases acid secretion. Somatostatin also acts on the G cells to inhibit gastrin release and the ECL cells to inhibit histamine release. Prostaglandins also inhibit acid secretion directly by decreasing the intracellular cyclic AMP and indirectly by acting on the ECL cells to inhibit histamine secretion. Gastric acid secretion occurs continuously, but between meals in the interdigestive phase, the rate of acid secretion is low because the very low gastric pH inhibits the release of additional acid through somatostatin. Then acid secretion is increased as we think about food in the cephalic phase, and even more acid is secreted as food enters the stomach in the gastric phase, and then entry of food into the duodenum allows only a small amount of acid secretion. We can use this cartoon to see how acid secretion is regulated during each of these phases. So in this image, there's a gland here in the fundus of the stomach that contains the acid-secreting parietal cells, the pepsinogen-secreting chief cells, as well as histamine-secreting ECL cells in the mucosa just outside the epithelial layer. There's also a gland in the gastric antrum that lacks parietal cells but contains regulatory cells, the gastrin-secreting G cells and the somatostatin-secreting D cells. Here in this resting or interdigestive phase, the pH in the stomach is very low. That's because there's a large amount of acid in the stomach but no food to buffer it. So this low pH or high acid content in the lumen directly activates the D cells, and they secrete somatostatin across their basolateral membrane. Somatostatin directly inhibits nearby G cells, decreasing gastrin production. And less gastrin means less acid secretion from the parietal cells. This negative feedback loop is a safety mechanism to prevent overproduction of acid while fasting. Now in the cephalic phase, there's no food in the system, but through the sight, smell, taste, or thought of food, the body is aware that food will likely be coming into the stomach soon, so it sends information out through the vagus nerve to prime the stomach and get it ready for a meal. Acetylcholine acts directly on parietal cells, causing them to secrete acid. And that vagal stimulation also causes release of the neuropeptide gastrin-releasing peptide, or GRP, which stimulates gastrin release from G cells. The hormone gastrin travels through the blood and acts directly on parietal cells to stimulate some acid secretion, but gastrin also causes histamine secretion, and gastrin and histamine binding together on the parietal cells acts synergistically to cause acid secretion. Pepsinogen secretion from the chief cell generally mirrors acid secretion, and vagal stimulation and acetylcholine release also stimulate secretion of that pepsinogen. The vagus nerve also inhibits D cells, reducing their release of somatostatin and therefore reducing the inhibition of gastrin release. The gastric phase begins with the entry of food into the stomach, and the presence of a meal in the stomach is what causes maximal stimulation of acid secretion. There are similar mechanisms as what we saw in the cephalic phase with vagal stimulation, along with a couple of other mechanisms. First, distension activates a vagovagal reflex. So gastric wall distension activates a vagal afferent pathway, resulting in a vagal efferent response. And just as in the cephalic phase, there's acetylcholine that's released that acts directly on the parietal cells to stimulate acid secretion. And the vagovagal reflex also results in secretion of GRP, which causes gastrin to enter the circulation. And that gastrin acts on the ECL cells as well as the parietal cells. In addition, distension also activates local reflexes. So local neurons release acetylcholine, which also acts on the G cells, causing gastrin secretion. And just like in the cephalic phase, the chief cells are also secreting pepsinogen. In addition, the presence of peptides and amino acids in the stomach also acts on the G cells, increasing the gastrin secretion even more. As chyme exits the stomach and enters into the intestine, the intestinal phase of digestion begins. So here, acid secretion is mediated by protein digestion components. The presence of protein digestion components in the duodenum stimulates some acid secretion, in part through their ability to activate duodenal G cells to secrete gastrin. So when chyme moves to the small intestine, the pH in the stomach drops. That's because there's no longer food present in the stomach to buffer that acid. So without a buffer, further secretion of these hydrogen ions causes the gastric pH to drop rapidly. And the presence of these many hydrogen ions in the lumen stimulates the D cells to secrete somatostatin, which in turn inhibits gastrin secretion. This chart summarizes the release of acid in each phase of digestion. The cephalic phase is mediated by the vagus nerve. The gastric phase is mediated by distension, which activates vagal reflexes as well as local reflexes, and it's also mediated by the presence of protein digestion components in the stomach. And the intestinal phase, which is mediated by the presence of protein digestion components in the intestine.